So today we can go ahead and get started. So we started talking about this process of inflammation. There's the other thing. I'm not going to post another set of slides for Monday. Basically, I made this set of slides, and I'm just going to finish it on Monday. So there won't be a new set. It's just this one for both today and Monday. So we talked about the beginning of this kind of general process of inflammation and how innate immune responses made by innate immune cells occur. And so we started talking about this process where we will have some kind of tissue damage allowing bacteria in. On Monday, most likely, I will tell you that this is sort of very classically what we think of as a antibacterial response. There is sort of a viral version of this, and we'll probably talk about that a little bit more on Monday. So this is very general, but the classic example here is usually bacteria. So I'm going to give you kind of the classic virus example later on. So we have tissue damage, we have bacteria coming in and being recognized by some cells at that site. As we saw last time, those cells are monocytes and macrophages that exist at that site. And those monocytes and macrophages are eventually going to do something, as this slide says. And in order to do that, they need to be able to recognize bacteria and know that there are bacteria present. Also remember that this whole process that we're talking about leads to sort of some of the cardinal signs of inflammation: redness, heat, swelling, and pain that you are all aware of. And we're going to see them come up. I'm going to make some comments about them a little bit later. So this process happens because of the ability of these macrophages to do something called self-non-self recognition. So and in fact, the general concept of self-non-self recognition is pretty much a major underlying concept of all of immunology. But here we have cells like our macrophage that have some proteins. Bacteria have some different proteins, and in fact also some other biological molecules. And our macrophage will have some receptors that can bind to those molecules. You can imagine those molecules, which are things that are unique on a microbe that are not found on our cells, as being microbial patterns, things that are often found on microbes. You'll see why I wrote it this way as we go. And so our macrophage will have lots of different types of receptors that can respond to those microbial patterns. And so these receptors are known as pattern recognition receptors, or PRRs. I will tell you that our innate immunity section of lab is also going to focus on some PRRs and experiments related to PRRs. That's what I do my research on in the lab. And so, you should be aware of what's going on here. These receptors are just sort of like normal old receptors. Again, that doesn't always make sense that I'm telling you that until later when we talk about adaptive immunity, where the receptors are not normal old receptors. So here they're just normal old receptors. The macrophage has many of them, so you can see lots of them expressed on this image of the macrophage on the right. And these receptors are really important because they are able to bind to conserved molecules that are part of many different microbes. So here you can see 3 different microbes, and you can see that they all have the same little shape. I don't know what you call that shape. And they all, even though there's 3 different microbes, the same receptor can detect all of them. So you don't have to have a huge number of these receptors to be able to detect a huge number of microbes. The idea is that they are responding to some conserved part of the microbial structure, which is great because A, it means that it's something that is going to be on lots of microbes, so you don't need to have a million receptors to respond to a million microbes. You can have receptors that are kind of hitting broad classes of microbes. It's also really important because that makes it very hard for the microbe to mutate and try to avoid or evolve around these receptors. This is like a major structural component of the microbe. It's not something the microbe is going to change very easily. Whereas, as we'll talk about later, sometimes with the specificity of the adaptive immune system, the microbe can kind of get around it. The microbe really is not going to be able to evade these in the same way. So we're looking at very conserved structures. We can see responses to broad classes of microbes. We don't really have to worry about microbes evolving away. And so those are some of the reasons why this is so really important for defense. The parts of the microbe that can be recognized also have a name. The name has actually changed over time in the field. So originally, this was known as, these were known as PAMPs. This table from your textbook gives you a list of some PRRs, or pattern recognition receptors, and the specific PAMPs that they recognize. So PAMP is also an acronym. The P at the end stands for pattern, because it's a microbial pattern. And in fact, note that the pattern is recognized by the pattern receptor. So these names kind of work together. Originally, PAMP stood for pathogen-associated molecular pattern. So the idea is that this is some structural feature of a microbe that is a pattern that we find in microbes and not in our cells that can be recognized. As we've learned more about PAMPs, we've realized that they are on a lot of different types of microbes, not just ones that cause disease. So microbes that are not pathogens have them too. And so we've started referring to them in the field more as something called MAMPs, which is microbe-associated molecular patterns. Like, we're not trying to call out what kind of microbe it is. We're being more vague. That it's just any old microbe. I am sure that I will use those words interchangeably because, well, I learned it as PAMP, and in my mind it's kind of still PAMP. But just so you know, in fact, microbes that are not pathogens can have these molecules too. The reason why I have this table here is so that I can show you some examples to help you kind of think about what a microbial pattern might be. So one example you can see is LPS, which is recognized by TLR4. LPS was really kind of the first one of these that was recognized. LPS is on the part of the cell wall of all gram-negative bacteria. And so this receptor is not going to recognize E. coli versus Salmonella. It's just going to recognize you're a gram-negative. And this is a structure that evolved, perhaps millions or billions of years ago. The microbe is not going to, in a week, make a new structure of it to get around this receptor. Similarly, we've got things like peptidoglycan, another part of the microbial cell wall. We've got some lipopeptides. We've got flagellin, which is the thing that makes up the bacterial flagellum. So basically any bacteria that has a flagellum gets recognized. We also have some nucleic acids in some cases. What's a microbe going to do? Change the entire structure of DNA to avoid this receptor? Like, that's not going to happen. So these are things that kind of are getting to a broad class of microbe, but also that are not easily changeable by mutation, so not easily avoidable. So these are kind of the big picture general idea of PRRs. I'm going to talk about PRRs as sort of general things right now, and then I'm going to come back and get into the specifics of some individual PRRs. So right now we just kind of care about PRRs as a general thing. And there are a couple of things that will happen to our macrophage when it recognizes a MAMP using its PRR. One of those things is pretty similar to things you've already seen, which is that if our macrophage, which has, you can see, many PRRs, that macrophage binds to a microbe using one of those PRRs, we're going to see phagocytosis of that microbe so that the microbe can be internalized and destroyed. And so phagocytosis is one part of this, but there's also another important part. Note that what I'm talking about here and what I'm talking about in the next slide are examples of things that go on with macrophages. So macrophages are phagocytic cells. One fact that a lot of people don't actually realize is, you learn about phagocytosis as a cell biology process. Only 4 kinds of cell in your body can do phagocytosis. It's not a thing that any old cell can do. There are only 4 phagocytic cell types in your body, maybe 3 even. One of those is, I'm being a little fast and loose with some definitions. Those are macrophages, neutrophils, and dendritic cells that we'll talk about later in the semester. And in today's class, as well as B cells, maybe. The reason why I'm pointing this out is that in today's class, both macrophages and neutrophils are going to come up. And both macrophages doing phagocytosis and neutrophils doing phagocytosis is going to come up. And sometimes people like to confuse them. They're like, oh yeah, those are the 2 cells that do phagocytosis. They think of them as being very similar. But in fact, the things that they do, they both do phagocytosis. That is true. They're both white blood cells. They both are part of the innate immune system. All that's true. They're both part of inflammation. We'll see them both today. All true. But they also have some things that are really different about them. And the thing that I'm telling you right now is all macrophages and PRRs. So macrophages can do phagocytosis and degradation when they recognize a microbe using PRRs, which are shown here as the little blue receptors on the cell surface. But macrophages also have almost what you could think of as like a bonus function. Macrophages also will do some signal transduction downstream of that pattern recognition receptor. Right now, that signal transduction is depicted as an arrow. Later, again, we'll talk more about the details of what's going on in that arrow. And this can change transcription in that macrophage cell. So this is, again, we're talking macrophages are doing this bonus function here. When this transcription happens, we start to have new RNA made, which of course leads to the production of new proteins, and those proteins can be secreted from the cell. And those proteins are known as inflammatory cytokines. Cytokines are important proteins that we see in the immune system. They are basically the proteins that immune cells make to talk to other cells. They're sort of the messenger proteins of the immune system. You can, in some ways, think of them as being like hormones. So this is how one cell talks to another. Here, we are seeing a particular type of cytokine. It's called an inflammatory cytokine because it's the kind that you make during inflammation. In the past, I have had students think that all cytokines are inflammatory cytokines. They're not. There are ones that do things other than inflammation. But in this case, we have the PRR leading to transcription, and then we get translation and secretion of our inflammatory cytokines. And so this is another big aspect of what happens following PRR getting activated. And those PRRs being activated is how the macrophage knows there's a bacteria and knows we need to start this process. And so you can see that we're going to see killing of the microbes. In fact, some of the phagocytosis will actually be enhanced by some of this signaling as well. We're going to see this inflammatory response that we're going to be continuing to talk about today. Some of this is actually going to help enhance the adaptive immune response as well. We'll get to that actually a bit later in the semester, how some pieces of this play into adaptive immunity. Right now you can know they play into adaptive immunity. We actually are realizing more and more, this is actually a pretty hot area of immunology, that we also can have like wound healing responses happening because of these inflammatory cytokines, which kind of makes sense. That, if you start to have you had that cut that got bacteria, and maybe you might want to turn on wound healing. Like, duh, that makes sense. Immunologists have, for years ignored that was a thing. And now we're suddenly like, wait a minute. One thing that's really cool, and so there was this paper that came out. I'd heard about it. I didn't really put a whole lot of attention into it. That was talking about one of the proteins we work on in my lab having a role in blood clotting. And I was like, blood clotting? That's like, what? Oh, well. And then COVID happened, and all these patients started to have weird blood clotting problems. And also COVID is associated with messed up innate inflammatory signaling. And I was like, wait a minute, inflammatory signaling could be related to blood clotting? So this is like a hot area that everyone in immunology is really excited about now, is the fact that there's also this tie-in, though we're not going to talk about it a ton right now. So what you've realized so far is that we can have this macrophage in our tissue that is going to recognize bacteria that come in. And it will release some factors, as we see here. Those factors are the inflammatory cytokines you just saw on the previous slide. They are made because the macrophage saw that bacteria using its PRR. And so this is an example of some of those molecules that are produced. And so really what we're talking about now is, okay, so we make these new things, these cytokines, and what the heck are they going to do? How do they actually do something about this microbe? So here are some specific kind of examples of this. You will note that my title here says cytokines and chemokines produced by activated macrophages. Chemokines are a type of cytokine. Officially, chemokine means chemotactic cytokine. It's a cytokine that makes things move. And so a lot of times they get lumped together, though technically chemokines are involved with movement. And so here you can see some examples of some of the cytokines that are actually made by an activated macrophage and what they do. And we're going to be talking about this more on some of the upcoming slides. So these include some cytokines like IL-1 beta, TNF-alpha, and IL-6. I usually in my brain, I often lump those three together, although they're not always lumpable together. But and these all have a lot of different effects. We can also see we've got a chemokine here, IL-8. And these together are actually going to lead to our state of inflammation. So we can see some of the effects of them here, and we'll also see them on some of the next slide. So one of the things we're going to see are some changes to the blood vessel. So changes to blood vessels in that area of the body. So changes to the vasculature. We're also going to see changes in metabolism in local tissue, as well as, because this is a chemokine, it makes things move. We're going to call in other cells. We're going to say, hello, cavalry, get over here. We got a problem. We need you. And so we're going to call in and recruit other cells. We're going to make some changes to the vasculature, and we're going to make some metabolic changes, all in this situation. I will also tell you, when I was an undergraduate, metabolism was sort of the bane of my existence. I was like, oh, that's the most boring thing ever. I'm so glad I'm an immunologist and never have to think about it again. One of the hottest areas of immunology right now is immunometabolism, and all of the ways that different immune cells actually mess with metabolism in their processes. And so I've had to take out my old biochemistry books and relearn metabolism, reteach myself metabolism. So any of these things that you think are like not relevant, they often become relevant later in your life. And so here we can see some of those effects of the inflammatory cytokines and chemokines, and again, specifically IL-1, IL-6, and TNF-alpha. And so here you can see on the left the before inflammation picture. And on the right, you can see the after. So before cytokine, after cytokine. So one thing you should notice, so we've got the tissue, so this is like the layer of skin, and this is the blood vessel under the tissue. First thing you should notice is that the blood vessel got a lot wider. So we've changed the diameter. This is called vasodilation. So we've had vasodilation where we've changed the diameter, and that's the first word up in that blue box. You can also look at the walls of the blood vessel. The walls of the blood vessel have become leaky. The cells are not as close together as they were before. That is called increased vascular permeability. So, and both of those changes to the blood vessel are the direct result of cytokines acting on them. So, why might it make sense that we make the blood vessel wider? Why should we vasodilate here? Yeah, Jamie.
A lot of immune cells are fairly large, and so to be able to get them to the site that they need to go to, we can increase the amount of, not only like the diameter so that they're able to move through it, but so that they're able to move faster.
Okay, so we want to be able to let cells move faster and get there. We also want more cells to be able to get there, right? And more blood. Because remember, there's some proteins in this blood too that might be useful, like say the complement proteins. And so we can get more blood and more blood proteins and more cells to the area. It's like we made the highway bigger. Makes sense, right? Why do we want the vessel wall to be leaky? Yeah, Justin.
I guess so that fluid can also be helpful.
Yeah, so that those cells and that fluid can get to the actual place where the bacteria are and be helpful. So we want all that stuff to be able to actually get there. We don't want a wall in its way. So we want to allow this to happen. So now let's think about this. You're going to have your blood vessel increase in size in a location of your body. And you're going to have it start leaking. What's that going to seem like to you as a person? What's that going to feel like in your hand? Yeah, or whatever. It's going to swell because more stuff's going there. It's going to get full of fluid and cells. So you put more stuff there, it has to get bigger, so it's going to swell. That's one of the cardinal signs of inflammation. That's specifically because of this cytokine response on the blood vessel. Yep, Rishi.
Oh, I was also going to say.
So heat, you say? Yeah, it's going to get hotter. The blood is actually warmer than some of the other tissues, and you're going to be moving a lot of stuff there, and we're going to have those metabolism increases I mentioned. So you're going to get heat. So another one of those cardinal signs of inflammation. Yep, Sebastian.
So when you get a mosquito bite, is that what causes the itchiness?
I'll come back to that. That's a really good question. I'm going to come back to that one in a second, okay? Because there's like 5 things I want to say about that. So what else? What is the other effect here? Is in fact also seen in a mosquito bite. Yeah. It gets red. As you're allowing more blood towards the area, you're going to get have that area start getting red. So 3 of the 4 cardinal signs of inflammation are really obvious here in terms of how the cytokine actually ties to the specific actions we're seeing. The 4th one is pain. And I'm going to wrap itch into pain a little bit. So it turns out that the cytokine, so this is a very simple immunologist drawing of this body location, but there are also going to be nerves there. As you'll see more and more throughout the semester, there's always this like battle between immunologists and neurobiologists. So we're not showing their stuff there. So there are some nerves there. Those cytokines can actually directly act on the nerve in order to induce the itch. There's considerably less known about either the pain or the itch piece of this, although it is actually a cytokine effect on the nerves. In fact, the world's expert on why cytokines make you itch and how cytokines affect nerves to itch was one of my classmates in grad school. And so like legit sat like next to me in like immunology class in my PhD. And so like every time I like now read like these things from this famous person, I'm like, that's just Isaac. What are you talking about? So that is something that that is exactly the type of thing he works on, or the itch piece of that. But yeah, the inflammation is why you're getting the itch, absolutely. So the other thing you could think about here, this is going to tie into something else I'm going to say, with the mosquito bite, is, goes back for a second. But this is fascinating. I get fascinated by a lot of this stuff, so sorry. So how do you start this whole business in the case of a mosquito bite? Like, what triggers the PRR to signal and make cytokines to give you that itch? Because remember, I'm telling you that the cytokines are the things that are actually leading to these effects. They make you get that little swollen place with a mosquito bite. They make you actually have the swelling and the redness and the pain and the itching. All the things of the mosquito bite are coming from this effect. And so in fact, they have to turn on inflammatory cytokines, and that happens with something engaging a PRR. So what the heck engaged a PRR in the case of a mosquito bite? Yeah, Justin.
I think it's probably because mosquitoes, like when they stick their little thing inside you, they release like proteins that make you not blood clot. Maybe your immune system might think that's important to you.
So one idea could be that it has to do with some of those proteins that are impacting blood clotting. It's actually not the same exact ones that are impacting blood clotting. But it's actually not a bacteria. It turns out you have a receptor for mosquito saliva. And so there is actually a PRR that responds to mosquito saliva to turn on this exact same process. And that will eventually lead to the itch because of the inflammatory cytokine production. Mosquito saliva and the immune system, fascinating. Yes?
So you know how when you get spider bites, they're bigger? Is that because we're more sensitive to whatever, I don't know, saliva?
So that is actually a slightly different type of immune response. And so that's a type of immune response that we'll get more into when, I think that really doesn't come up until we start to talk about allergy, so it'll be later in the semester. But that's closer to an allergy response than the mosquito. Awesome. Okay. So I have my slides here in front of me. I don't like the order of the next slide, so I'm going to move it. I'm going to skip it for a second. So one other thing that happens here is that we are going to be specifically recruiting in other cells to help clean up here. And so you can even see that here where we see a few other cells that look a little different than this macrophage coming to the area to try to help out. And that recruitment, that call is also happening because of those inflammatory cytokines. And so you can see we do have this recruitment of other cells. In this case, those other cells are generally neutrophils. And so we call in a whole bunch of neutrophils to come to that site so that the neutrophils can start to clean up the bacteria that have gotten in and potentially divided in that location while this whole thing was going on. So they are also recruited by some of these different molecules. So just to go back to that slide I just skipped, hopefully you kind of get an idea of how inflammatory cytokines, and in particular the chemokines, will work. If you think back to things we learned about last time with complement, C3a and C5a basically act as inflammatory chemokines and inflammatory cytokines. So I told you that we have these random complement proteins that are part of the protein part of this whole business. They actually act the same way as what we've just seen with these cytokines. So this is another place where we can sort of artificially make these separations and say this category is like this and this category is like this, but they blend together. C3a and C5a specifically change permeability of blood vessels, make the blood vessels leaky, and make cells migrate to the site. So C3a and C5a are actually doing this exact same thing. They have a receptor that looks almost exactly like a chemokine receptor, and it's really just this sort of exactly the same thing that you see with these chemokines and cytokines. So realize it's just a variation on exactly the same theme that you're seeing. And so all of this information of what we're talking about is known as local inflammation. And so you can see that example of local inflammation here. And so what you should notice is that this person had something happen to their hand, and they get inflammation in their hand. See, it got red. And that means that the blood vessels in their hand had changes happen to them in terms of their diameter, their permeability. We have more cells come in. And that's good. We got rid of that infection. The hand is better. So they survived. We also are going to turn on the adaptive immune response. So notice that when this inflammation is happening, these blood vessel changes and the changes that we kind of think about as those inflammation effects are only happening in that very same location of the body, locally. We're not seeing them everywhere else. So the rest of the person's body is not red. Yep.
So, when you see swelling when there's a local infection,
Yes.
we also see inflammation when, like for example, you shut your hand at the door. So how is that? It doesn't seem like it's the same.
That is a fantastic question also. And that's another thing I want to bring up here. So usually the example that I give is when you sprain your ankle, right? You sprain your ankle, sometimes your ankle swells and you have injury. Imagine that like the skin didn't break, nothing happened, or the same thing, you slam your hand in the car door. I dropped an incubator on my finger on Monday, no microbes in any of those cases. Yet in all of those cases, we see the same 4 cardinal signs of inflammation. The heat, the redness, the swelling, the pain. And so again, you might look at what I'm saying and say, that's a time that I've experienced local inflammation, Dr. Barker. I don't buy all the stuff that you're telling me right now, because this is a different issue. So this is also a super hot area of immunology. So officially, this is known as sterile inflammation, inflammation when there's no microbe. And what we know about sterile inflammation is that PRRs are still involved, but they don't have a microbial product that they are recognizing. Instead, they recognize a different molecule known as a DAMP. And the DAMP is depending on which immunologist you talk to, either a damage or a danger-associated molecular pattern. So when you have a lot of cell death and when you have a lot of cell injury, you actually get some product made by the cells that can hit certain PRRs and lead to this same process. Exactly what those products are, exactly how this works, is a very hot area of research. And so what would be happening in that situation is that you would actually have damage to the tissue when you slam your hand in the car door, and the cells that experience that damage would make some of these products that would stimulate PRRs and lead to this exact same process. So it is actually the same process, it's just a different ligand starting it off. Like I said, some people call it damage, some people call it danger, and the details of it are very much up in the air. I will also just tell you that in the past, so I actually went to a whole conference on DAMPs in the fall, or in a past time. All the times blends together now. I don't know when that was. In the past year. There was this hypothesis in immunology in the 90s called the danger hypothesis and danger signals. A lot of people it kind of went out of favor. A lot of people stopped talking about it. In fact, danger is one of two words that I say in immunology in this class that I'm afraid like lightning is going to strike me down. Because when I originally learned immunology, it was like, that's one of those words you never say. Like, we don't believe in that anymore. And so that's why sometimes people call it damage, because they don't want to say the dirty word. But more and more people are starting to accept danger, and I think danger is a nice way to actually talk about what we mean now. And lightning, again, did not strike me after all these years. So does that answer your question? And yes, there's lots of cool stuff about what's going on with DAMPs and DAMP signaling. So we were talking about local inflammation, which can happen in the case of microbial infection and MAMPs, or in sterile inflammation with DAMPs. But there are some changes out to other parts of your body that will happen when you start to make inflammation in a location like your hand. So some of those cytokines will travel to the liver. And when those cytokines, like IL-6 that you can see here, travels to the liver, it will result in the liver making some new proteins. So we'll get new proteins made in the liver. These liver proteins are known as the acute phase proteins. Some of them help blood clotting. It makes sense. Now we're going to try to increase the amount of clotting we're going to do. One of the other big groups of proteins that get made here are complement proteins. So now you've started this inflammatory response. You say to the liver, hey, can you make me some more complement proteins in case I need to do more complement activation? So again, even though I can separate the cells do this, the proteins do this, you can see ways that they are interlinked. And there are a number of other proteins that are getting made here by the liver. So we have this acute phase response. We also see some of those cytokines act on the bone marrow, where we have been storing a whole bunch of neutrophils. And this says, hey, bone marrow, please release all the neutrophils. We need a lot right now. And so those neutrophils that were stored in the bone marrow get released so that we can have a ton of neutrophils ready to come to that local area. And so even though the bone marrow is not necessarily the thing that is the inflammatory signal, it's getting an effect. There also are some other things that are going to happen. Some of those cytokines are going to work in the brain at the hypothalamus and on fat and muscle tissues, all of which are going to increase body temperature and also change some metabolism, which is kind of going to impact body temperature. This is important because in many cases, microbial cells are not going to be able to reproduce as well at higher temperatures. So this is going to kill them or make them stop replicating. There also has been a fair amount of data recently that have also shown that many of our immune cells work really well at high temperature. So it's basically like giving our immune cells an advantage and giving the microbes a disadvantage all at the same time. There's one downside to this. What's a potential downside to increased body temperature? So we're making increased body temperature. Yeah, Sydney? Our cells are not awesome with a lot of heat. And so if this were going to go on for like ever, for a really long time, it's going to start to damage our cells. So we need to sort of turn up this temperature quickly for kind of a short period of time, stop the microbes, help the immune system, and then go back to normal. There is another word for this part of the process, and this thing that inflammatory cytokines do. What is our other word? Yes? Fever. So in fact, these inflammatory cytokines induce fever. And this is the fever response. And so you can see this here. These inflammatory cytokines are going to act on the bone marrow to change how many cells are coming out of the bone marrow. They're going to act on the liver to make different proteins from the liver. They are also going to act on the brain to induce fever. They may sometimes also induce fatigue. There are also situations where many of these will be able to do things like make you lethargic, give you muscle pain, lower your appetite, lead to nausea. There's also a great symptom that sometimes comes up here. You know the general blah feeling when you feel sick? That's called malaise. And malaise is also an effect of inflammatory cytokines. So all of these symptoms and all of these things that you feel in your body are the effects of inflammatory cytokines. This is a super important concept in immunology. So realize, oftentimes you feel these things and you're like, oh my gosh, I feel sick. I can't believe the microbe did this to me. Microbe didn't do it to you. You did it to you. In order to give you some physiological advantages to help you combat those microbes. And so generally when you are feeling these symptoms, it means your immune system is doing something. It means your immune system is trying to fight a microbe. You gave yourself a fever. Your cytokines gave you the fever. The microbe didn't do that to you. And so sometimes if you think about examples of times when you might feel some of these things, here's one of the many thousands of times I like to get on my soapbox about this. For example, if you say got a vaccine and felt some of these things, the answer is, yay, that means your immune system is making an innate immune response. And the cells of the innate immune response are doing their job. And whenever any of my friends complain about I got a vaccine and now I have a fever on Facebook, I always reply, I'm so happy to hear that your immune system is working as it should, and none of them have punched me yet. So this is kind of what that inflammatory response should look like. So at this point, we have had bacteria come in. We've had a macrophage recognize those bacteria. We've had, using its PRR, we've had that macrophage do some transcription in order to make some inflammatory cytokines. We've called in our neutrophils, as well as some other proteins and other fluids, and those antimicrobial peptides that we talked about earlier. All of them are coming in to help us get rid of the microbe. And so now the last thing we've got to do is we actually have to kill some bacteria. And we actually have to get rid of this microbe and finish this process. And this is really done by the cells that come in here, which are the neutrophils. And so this is, again, why this is, if you remember, I told you that this whole process is sort of like 4 to 96 hours, not immediate, because we need to do some transcription and translation and then call in the cavalry and have the cavalry actually show up before we are able to finish up this process. And so now we're going to see the neutrophils acting on these microbes. And the neutrophils are largely going to work by phagocytosis. So with phagocytosis, the neutrophil will bind to that microbe using some kind of receptors, and this will actually result in that microbe being inside of a compartment in the cell. So the cell is actually wrapping some of its membrane around the microbe, and that puts the microbe into a little compartment surrounded by membrane in the cytoplasm. Because that compartment didn't exist before this, it was made through this process of phagocytosis, we call the compartment a phagosome. It's an organelle that was made by phagocytosis. So the bacteria is ingested, and that forms a phagosome. The phagosome is then going to fuse with some other organelles in the cell. Specifically, those organelles are going to be lysosomes, which are at much lower pH. They're very acidic. And that between the pH here, as well as the some enzymes that are only active at that low pH, we're going to degrade that microbe to the point where it is basically going to be like short amino acids and small things that the cell can use for metabolism now and your body can use for metabolism. If you remember, well, here, I'll show this to you on the next slide as well. So here you can see another version of this, where our neutrophil is going to perform phagocytosis and make a phagosome. And that phagosome is eventually going to actually fuse with the lysosome. Sometimes we then call it the phagolysosome, because we're clever. But what you also might remember from lab 1 is that we called neutrophils a type of granulocyte, because they had lots of granules. You can see those granules here as different colored shapes. Those granules will fuse with the phagosome and lysosome as well, and basically deliver a whole bunch of peptides and enzymes to kill microbes. So that's what the point of those granules is in the neutrophil, is to turn on this response so that we can then completely degrade that microbe. One of the ways that we can degrade that microbe is through a process known as respiratory burst. So inside of that neutrophil, we will see some enzymes like NADPH oxidase, superoxide dismutase, myeloperoxidase, that take oxygen and turn it into some other compounds. You don't really have to care what the other compounds are, but I will point them out just for your sake, your knowledge sake. One of them ends up being hydrogen peroxide. You know about hydrogen peroxide. You know it's a thing that you can use to kill bacteria, put on a wound. Yeah, so the macrophage makes, or the neutrophil makes that to try to kill bacteria. Similarly, you can see we get some hypochlorous acid. This is the active agent in bleach. And so basically inside the neutrophil, we're making these compounds that you know about that are going to kill those bacteria. As a group, all of these compounds are known as the reactive oxygen species because they all have oxygens in them. There are some similar compounds containing nitrogens known as the reactive nitrogen species that are also made with the ability to kill microbes. Nitric oxide is the one that is sort of the most famous and that people spend the most time studying. So the neutrophil is going to specifically start making ROS, reactive oxygen species, and RNS through this process of respiratory burst in order to kill those microbes. In addition, those microbes are actually full of antimicrobial peptides. So many of the antimicrobial peptides we talked about before that act outside of cells also live inside of macrophages. So and this is another place where everything we're talking about kind of ties together. And so those macrophages and neutrophils have lots of antimicrobial peptides in order to kill anything they might have phagocytosed. So I've got a couple of other fun things to think about in terms of phagocytosis. One of them is going to be a quick diversion, but I feel like it's an important diversion. So this slide, this figure from your textbook, shows you an image of some details of phagocytosis. But I also, and one thing that you can notice is in the top, it involves actin. It actually involves a fair amount of work on the cell's part, a fair amount of cytoskeletal rearrangement. I mentioned before that only a few cell types in your body have the ability to do this, and it does actually take a lot of work for the cell. So if one has a working computer, and if one ever goes to YouTube and looks up, well, it's probably better to do it this way. Neutrophil phagocytosis. You can find a video. This is a very famous video that all immunologists know.
Are white blood.
We've all seen it about 1 million times. Sometimes I root for the bacteria because I've seen it so many times. So here, and you can also find this video set to any music you want it set to, including things like the theme to Cops and the theme to Benny Hill. Apparently, there are some people who have too much time on their hands. In any case, what you're going to see here is we're going to be able to actually watch a neutrophil do this process. What you will notice is actually in this prep that they've made, there are all of these black cells. Those are actually red blood cells that were contaminants in the process. They don't matter at all for the point of this, although it ends up making the video fun because it looks like they're obstacles. And you're also going to see a bacteria, which will be pointed out, which is a Staphylococcus.
Hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
So what I hope that you notice there is first of all, a lot of the movement of the neutrophil, which is largely happening via some of the PRR signaling that we're seeing. You can see how much dramatically, when that neutrophil does phagocytosis, it changes shape pretty dramatically. There's actually a lot of cytoskeletal and cell biological movement that the cell has to do. And you can see that there is kind of this directional work that's going on. If you actually watch that video closely, at one point, the neutrophil turns and goes right by a bacteria and like misses it because it's going after the one it was chasing. I think it's right, it's about here. So.
Chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized.
See, look, it missed that one.
Chasing the bacteria. The bacteria bounced around by thermal energy.
And that's because there's so much signaling going on sort of in this direction after that one bacteria that it's chasing that it can't really just like pivot and be like, oh, actually I'm signaling over here now. And so there's a lot of this going on. So first of all, I thought it was important that you see this phagocytosis in action and not just, as a cartoon, because that's much more fun. Yeah, I really like that video. It's a good video. So the other thing that we need to talk a little bit about is what happens after that phagocytosis with the neutrophil. So here you can kind of see this process of the neutrophil does phagocytosis. Here's the one I want. So the neutrophil does some phagocytosis, degrades the microbe all up, down to its component parts that can be used for metabolism. But then we have to think about what happens next, and there is actually one more step that happens after this. And in fact, the thing that happens after this process is that the neutrophil dies. So neutrophils are very, very short-lived cells. Basically, once they do this phagocytosis, once they use boatloads of energy and metabolic processes to do respiratory burst, once they use all this energy to chase bacteria around, then they die. And so those neutrophils will die, and they will eventually get phagocytosed by macrophages, who you can kind of think of as the cleanup crew. So they're going to clean up the corpses, and they're actually going to be the cells that are using all of those carbohydrates and nucleic acids and amino acids that came from this degradation. You actually have probably seen dead neutrophils before in your life. Remember, white blood cells are white. Neutrophils are a type of white blood cell. And when you have some kind of inflammation, you'll get a lot of neutrophils there, and then you'll get a lot of dead neutrophils. So many that you can see them, and you can get a little area of them. A group of dead neutrophils is known as pus. Probably heard of pus before. That's just dead neutrophils. That's all it is. What I will also tell you when I put together this lecture many years ago is I learned an important lesson, which is do not ever do a Google image search for pus, because you will see lots of things you did not want to see. So that's kind of like the original story on this, but there's one additional feature to all of this that's super cool that immunologists have also learned more about. So it turns out that there's some more complexity to what goes on when the neutrophils die. Just like I told you that macrophages have a special bonus function, which is that ability to do transcription and make inflammatory cytokines, neutrophils also have a special bonus function, and it has to do with what happens to them when they die. And so when neutrophils die, they will sometimes form these structures known as neutrophil extracellular traps. And neutrophil extracellular traps is often abbreviated as nets. People love to call neutrophil extracellular traps neutrophil nets, which is redundant because the N already stood for neutrophil. So we see a lot of neutrophil nets, or this process of cell death called netosis. So you're making this net as you die. These nets are actually big collections of DNA and histone. So it's the chromatin of the cell. It's DNA and some other proteins that the neutrophils basically throw out when they're dying. And they throw out these nets because they're sticky. Neutrophils are basically Spider-Man. They throw out a whole, basically as they're dying, they're like, I don't need this DNA anymore. Let me throw it out as a sticky net to catch any bacteria that might be around. And you can see in this figure from your textbook, here is chromatin from neutrophil nets trapping a whole bunch of E. coli bacteria. Here you can actually see a net being extruded from a dying neutrophil. So neutrophils have this ability to make nets that also will be able to physically trap microbes. Again, this is going to help us keep our numbers down if we just get rid of those fast replicating microbes, and this will help get rid of them. So as we mentioned last time when we talked about inflammation, inflammation is a super important physiologic response that we have to deal with infections and tissue injuries and other types of stress. And it's a good thing. But in many places you hear about inflammation like it's a bad word. It's a very bad thing that has to be avoided. And there are also bad sides to inflammation. If we have inflammation in a way that is not regulated, so too much, or in the wrong place, or too long of a period of time, then it goes really bad. And that can lead to autoimmunity, tissue damage, sepsis, fibrosis, tumor growth, lots of different types of diseases, all sorts of bad news. And so I just want to show you a couple of examples of this process. The most famous one is something called septic shock. In septic shock, we have some molecule, some PRR, that induces inflammation not in one local site, like in the hand in the previous example, but body-wide. The most common example here is if you have a bacterial infection in the blood, you've got PRRs everywhere, because they're everywhere throughout the blood. And so all of the blood vessels are getting hit by this inflammation process instead of just some at a local site. So all of your blood vessels are going to increase in diameter. All of your blood vessels are going to become leaky. Let's think about this for a second. If all of your blood vessels increase in diameter and they all become leaky, what's going to happen? Yeah, Jamie. Your blood pressure is going to drop. Why? Let's make sure we're very clear on what's going on.
Because of the amount of vasodilation happening, your heart just can't keep up with the volume that it needs to produce.
So A, the pressure is going to change because of the diameter of the vessel, but there's also another piece to that, which is that all the blood leaks out of the vessels because they're leaky now. So all the blood stops being in the vessels and starts being in all the random tissues. So you actually have problems with your blood volume and the amount of blood you have because it's not being in the blood anymore. It's outside in the tissues. As a result, you have swelling at your whole body, which is systemic edema. You have problems with blood volume. You have the wrong numbers of proteins and cells in your blood, and you can actually have your blood vessels collapse because there's nothing in them to help keep them open. You also will then start to have issues with coagulation, and you can actually lead to organ failure and septic shock. This is actually one of the most common causes of death due to microbial infection. This is a major issue in hospitals. And this is one of those things that I think a lot of people would want to kind of try to cure. And so understanding inflammation, understanding, say, the signaling that can lead to some of these inflammatory cytokines being produced is really an important aspect of understanding this process and why immunologists care so much about this. Yep, Justin.
So if someone has septic shock, like, do they just give a medicine to stop the cytokine or just let it run its course?
So usually they do two things. One of them is they give them fluids actually to deal with these volume issues, and so they're trying to give them more fluids. And typically they will go with something like an antibiotic to try to get rid of the bacteria or do something to stop having the cytokines get produced. In general, cytokines are produced for relatively short periods of time. So if you can stop the production, then you'll kind of can hopefully clear this up. You also might have to deal with some of the other sort of anatomical and physiological changes that happen as a downstream result. But fluids and antibiotics are sort of the first round on this. And we are going to talk a little bit more about a couple more bad sides of inflammation, as well as moving into the virus piece of this and the signaling piece of this on Monday. So I will see you guys then.